Welcome to the continuation of Nutrition in Animals 2. So there are 2 more videos to go, so just enjoy. So just a recap of what we discussed in the previous video. We were looking at the digestion of food substances along the alimentary canal. The food substances we concentrated on carbohydrates, proteins, and fats or lipids. So up to now, this is what we have discussed. We discussed what happens in the mouth. We said in the mouth there is chemical digestion and physical digestion. Chemical digestion is focused on the carbohydrates. What carbohydrate? Starch. So during chemical digestion in the mouth, starch is broken down to maltose by an enzyme called salivary amylase. That's the only chemical digestion that occurs in the mouth. We also discussed of the physical digestion whose purpose is to increase the surface area. That's why the food is broken down. We did discuss some other important some other important facts which were centered on the components of saliva, such as water, the enzyme zymase, the alkaline temperature in the mouth. So those are very important. Then from there we went on to talk about peristalsis. When you swallow the food, how does the food move till it reaches the stomach? Then after that went into the stomach. So in the stomach we also discussed some food substances that are digested there. So we talked of proteins. Okay, we did highlight that proteins are broken down to form peptides by the enzyme in the stomach. Okay, that is pepsin. Then we also talked about sucrose, which is broken down to glucose and galactose. Okay, so this breaking down does not involve an enzyme. It's just the hydrochloric acid which hydrolyzes sucrose. To hydrolyze is more like to break down using water, or to break down by more of dissolving something so that it breaks into its constituent parts. Okay, so one thing also mentioned is in the mouth the digestion of carbohydrates is not completed. The digestion of starch is not completed. Why? Because food does not last long in the mouth. When we're chewing, we swallow, so part of the carbohydrates is digested well, the other part is not digested. So where does the digestion occur? The second digestion, when starch is digested partly, where is the rest of starch going to be digested? So we hope to answer that in this video. Also, proteins are not all of them digested in the stomach. And finally, we haven't talked about fats. Where are they digested? So those are the key important questions we should have in mind as we go on with this video. Another interesting fact is let's think of how far we have gone so far. We learned about enzymes. We talked about naming of enzymes, how to name enzymes. Okay, what's the importance of that information other than passing the exam? The importance is it will help us understand the rest of biology. So we're going to see the application of that knowledge on how to name enzymes. If you have forgotten, you can quickly just go into your book, just revise just a bit to make sure that you remember that if an enzyme works on lipids, it's going to be called lipase. Okay, and if an enzyme works on sucrose, it's going to be called sucrase or sucrase. So those are important facts to remember. And also there is more information as we are going to see here. There's a lot of information, but that information is not new. It's the information that we already know. So most of the things are going to be more of a revision, reminding us of what we studied and how they connect now with everything about digestion. So we're going to continue on our journey from the stomach to the small intestines. So from the stomach, we go to the small intestines. Small intestine starts from here and they move up to here. So these small intestines have 3 parts. There's the duodenum, then there's the jejunum, and there is the ileum. So digestion occurs in the duodenum and in the jejunum, but not in the ileum. Okay, no digestion occurs here in the ileum, only in the jejunum and duodenum. So just like in the stomach where there is gastric juice, and in the mouth where there is saliva, there are some juice that helps in digestion in the duodenum. So the first kind comes from this structure here, which is yellow in shape, and it is behind the stomach, called the pancreas, which secretes pancreatic juice. This pancreatic juice is taken to the duodenum through a pipe or a tube called the pancreatic duct. So if you go to that picture which I'd shown in the previous video, or those with notes, in the notes you're going to see that there is a tube connecting the pancreas to the duodenum called pancreatic duct. Another type of juice that helps in digestion here comes from the liver. So the liver secretes what is called bile. This bile is stored in the gallbladder, and when needed, the gallbladder is going to push the bile through the tube here called the bile duct into the duodenum. Okay, so in the duodenum there's going to be bile and pancreatic juice. Okay, so the question is, what does bile do and what does pancreatic juice do? So to summarize what I've said, let me just be writing them. So the duodenum receives digestive juices from the liver and the pancreas. The liver secretes bile, which is temporarily stored in the gallbladder and carried to the duodenum by the bile duct. Bile contains three components. The first is called sodium hydrogen carbonate. Sodium hydrogen carbonate. The second are called bile salts, and the last is called bile pigments. So bile is made of sodium hydrogen carbonate, bile salts, and bile pigments. The question is, what do each of these do? So I'm going to start with sodium hydrogen carbonate, which neutralizes the acidic chyme and then sets an optimum alkaline pH for the enzymes. So if we go back before we reach the duodenum, digestion occurs in the stomach. So everything in the stomach, that is the undigested food, the digested food, the gastric juice, is going to be taken to the duodenum. So the gastric juice, remember, it's acidic. Therefore, the entire mixture is going to be acidic. Now, for some of the components to be digested, they need an alkaline pH. Remember, we said in the mouth, starch is not fully digested by amylase. Okay? Because of that, it has to be digested somewhere. And again, we say that in the stomach, it is not digested because the environment is acidic, therefore amylase cannot work. So it is in the duodenum where starch is further digested. But for that digestion to occur, the pH has to be alkaline, not acidic. Okay, so this sodium hydrogen carbonate makes sure that the acid is neutralized. To neutralize the acid is to remove its acidic property so that it has the same property as water, so to put it. Okay, so it makes the acid it makes the acid lose its acid property or acid characteristics. Okay, so that's what to neutralize is. For those who still can't get it by the explanation that I've used, in case you have been bitten by some insect, sometimes you can put some things there, some herbal, to neutralize the poison. Okay, so even here, the sodium hydrogen carbonate neutralizes the acid so that it loses its acidic property. The chyme, remember, is the mixture of everything that comes from the stomach. Additionally, this sodium hydrogen carbonate makes sure that the environment is alkaline so that enzymes such as amylase can further work on starch. The bile salts emulsify fats, thereby increasing the surface area for the action of lipase. So remember what we discussed on enzymes, the way enzymes are named. Just from the name of this enzyme, you can predict or you can tell what its substrate is. Lipase, lipids. Remember? Okay. So what is to emulsify before we waste time on lipase? To emulsify is to break down large fat droplets into smaller ones. It's more like chewing, which happens in the mouth, where large food particles are broken down to smaller ones. But emulsification is focused on fats, breaking them into smaller ones, with the same purpose as chewing, that is to increase the surface area. So emulsification is the breaking down of large globes of fats into smaller globes. With what purpose? To increase the surface area. Then the third one, or the third component, is the bile pigments. So these bile pigments do not have any digestive function but add color to the feces. So they don't do anything, they just add color to the feces. That's their job, but they do not help in any way in digestion. Okay, so sodium hydrogen carbonate to neutralize and add an alkaline pH. Bile salts to emulsify fats so that there is a large surface area for digestion. Bile pigments, no function. So now we go to the pancreas. So the pancreas secretes a juice called pancreatic juice. So the pancreatic juice has the following components. First, it has sodium hydrogen carbonate, which is the same sodium hydrogen carbonate that is found in bile, and it does exactly the same thing. Then there are three enzymes that are part of the pancreatic juice. The first one is called trypsin. The third is called lipase, which would expect to work on lipids. Then the last one is pancreatic amylase, which is the same amylase as the one secreted in the mouth, doing the same function. The difference is this one comes from the pancreas and the one in the mouth comes from salivary glands. That's the difference. This one comes from the pancreas, then that one comes from salivary glands. So let's take a look at the function of each of the components. But for sodium hydrogen carbonate, it's the same function, that is to neutralize and to add an alkaline pH. Then trypsin breaks down proteins to form peptides. Okay. So what else does this? It's pepsin. In the stomach, there's pepsin, which breaks down proteins to form peptides. But in the duodenum, there's trypsin, which does the job. The third is lipase, which breaks down fat molecules to fat acids and glycerol. So these fats have been emulsified by the component in bile, which are bile salts. So once they are emulsified, digestion becomes easy, or it is easy for lipase to work on the emulsified fats. Okay, so they are going to be broken down to their building blocks. Remember what we studied on nutrition. Fatty acids and glycerol makes up fats. So lipase does this. It breaks down fats to form fatty acid and glycerol. So even this peptide, at some point, it has to be broken down to amino acids because this is a protein. Then the pancreatic amylase breaks down starch to form maltose. So the starch that was not digested in the mouth is dealt with in the duodenum. So once digestion is done in the duodenum, we go to the jejunum. So in the jejunum, there are what is called intestinal juice. So this intestinal juice contains 4 enzymes: lactase, which works on lactose; maltase, which works on maltose; sucrase, which works on sucrose; and peptidase, which works on peptides. Okay, so let's just go through each of them. So number 1, lactase breaks down lactose to glucose and galactose. we know that lactose is a sugar which is made of glucose and galactose. We did this on Introduction to Nutrition. Okay, but where does it come from? So this lactose is found in milk. In milk there are two nutrients. There is a sugar and a protein. Okay, so a protein is the one that is dealt with in the stomach, okay, by pepsin. Then what remains in the milk is lactose. So lactose is dealt with in the jejunum. Okay, so lactose is going to be broken by lactase to form glucose and galactose. Then maltase breaks down maltose to glucose. This maltose is what was formed when starch is broken or is digested. Starch in the mouth was digested to maltose, and also here in the duodenum it was digested to maltose. So the maltose is going to be digested further to glucose. So you can see that the pattern here is we want to break everything into their building blocks, because building blocks are the ones that the body is going to use. Okay, so we want to make sure that proteins are changed to amino acids, then sugars or carbohydrates are changed to their simple sugars, okay, and lipids are changed to fatty acids and glycerol. So the third is sucrase, which breaks down sucrose, which is also a sugar, to glucose and fructose. We also discussed this, that sucrose is made of glucose and fructose, when we did Introduction to Nutrition. The last one, peptidase, breaks down peptides to amino acids. Hence, digestion is done. Everything has been broken down. Okay. So fats are dealt with here in the duodenum, yes, in the duodenum, not in the jejunum. Okay, then in the jejunum, carbohydrates are dealt with and proteins are dealt with. So in summary, in the mouth there's only carbohydrates which are broken down. In the stomach, that's where we see carbohydrates and proteins. Then in the duodenum, we see also fats are being digested, and here is where the final digestion occurs. So now we go to the ileum. So the ileum doesn't do any digestion, it just absorbs those things that have been digested. It's going to take them into the bloodstream. Remember the stages of Holozoic nutrition: ingestion, digestion. So we're done with digestion, we're now on absorption. Okay, ingestion is just when you put food in the mouth. Then digestion is everything that starts from the mouth up to the jejunum. Then now we are absorbing the food so that it goes with blood and is taken to the cells that need that food. Okay, so how is the ileum adapted to to its function? So before we can discuss the ways in which the ileum is adapted to this function, let's take a look at a very important structure found in the ileum called the villus. So this is a villus, plural is villi. That is if they are made, they are called villi. Yeah, so these are like they are finger-like projections found in the lining of the small intestines, especially the ileum to be specific. So they are the ones that have that are tasked to absorb food substances. Okay, so when you look at this villus, on top here it is covered by the skin of the inside of the intestines. So inside the intestines there's a skin covering the intestines, so it is this, so it keeps on going, then there's another one just like that. Okay, so this skin is called epithelium. Okay, so for those who are interested in knowing maybe where the word comes from, epi means upper. Okay, and maybe the other part comes from the fact that this is an ileum. Okay, so it's like the upper skin of the ileum. Okay, In short, just master it to be epithelium unless you're interested just for remembering purposes. So the top part is called epithelium. Then just underneath the epithelium, we have blood capillaries. So these blood capillaries are the ones that are going to get the absorbed food and they'll take to the various parts of the body. Then on the middle of the blood capillary, there's what is called lacteal. So this lacteal absorbs fatty acids and glycerol. So blood capillaries will take other substances, but fatty acids and glycerol are taken by the lacteal. Okay, so now let's take a look at the features that makes the small intestines or the ileum adapted to its function. So the features, of course, will address the presence of this villi and this epithelium. So the first is the ileum is very long, thereby providing a large surface area for absorption. Since it's very long, so as the digested food is moving, it will be absorbed slowly and slowly. So scientifically we say it provides a large surface area for absorption. 2, it has a thin epithelium for more efficient diffusion of food. So the epithelium is thin so that food can easily penetrate it and go into the bloodstream. 3, it has finger-like projections called villi and microvilli. There are two types. There are villi and microvilli. Microvilli are small, which further increase the surface area for absorption. Okay, so the fourth one is each villus has a network of capillaries for absorption and transportation of monosaccharides and amino acids. So I showed you under the epithelium there were capillary networks which are going to absorb sugars and proteins in form of monosaccharides and amino acids and then transport them to other parts of the body. Okay. Then finally, each villus has a lacteal which absorbs and transports fatty acids and glycerol. So remember, the first time you encountered this word villus was when we were doing active transport. Okay, so don't forget that. So most of the things that we learn them, and at some point we might come back to them. Okay, so the way food substances are absorbed, of course, there's diffusion and there's also active transport. Okay, so these are the features that makes the ileum be adapted to its function. So this marks the end of part 2. So part 3 is going to just summarize or look at some of the important information about the alimentary canal, such as the function of the liver and some of the common ailments of the alimentary canal. Then from there, we'll conclude this topic by taking a look at dental formula. Happy solving.